Looks like we've got who we're going to get, and if some other folks come in a little bit later, that'll be fine as well. My name is Bob Olson, and I'm going to, on behalf of the WSU Emerit Society. Good man. As soon as I announced that there were more coming, there are more coming. Good. Yeah, on behalf of the WSU Emerit Society and the Nuclear Science Center, I'd like to welcome you to this lecture by Dr. Tony Brooks. He's an emeritus faculty member from WSU Tri-Cities, and this lecture is entitled The Science of Radiation Biology and the Fear of Low-Level Radiation. Now, in addition to his position at WSU, Dr. Brooks' career included several other stops, but one as a laboratory senior scientist and section manager at PNNL. His research interests included cytogenetics, radiation-induced cancer, radiation risk, and public outreach on radiation effects. And specifically, he worked on understanding the biological changes induced by low doses of ionizing radiation. He served also as the principal investigator and chief scientist for the Department of Energy project, DOE Low Dose Radiation Research Program, summarized in his book, which was published by WSU Press here, Low Dose Radiation, the History of the U.S. Department of Energy Research Program. Now, in my opinion, it's rare that scientific research has the potential for significant impact on society, but this work really does raise important questions about the method that we now use for evaluating the impact of exposure to ionizing radiation. And the method that's used now is what's called the linear no threshold theory. And there are some significant questions that are being raised about this, and Tony will talk about that. But in my opinion, this research is some of the most important in recent years because of that. I think the lecture is timely, in part because the subject of nuclear power is being raised and discussed by many people, among others, the artificial intelligence community. And I think you've heard about this in the news recently, where they're talking about the amount of energy that they need and some of their plans to use small modular reactors, for example, to get this energy and for these artificial intelligence projects. And so we're hearing more about that. And yet, at the same time, there is a lingering fear among the population that must be addressed. And I think Dr. Brooks is well qualified to do this. Now, I might add that in case you have further interest in this, Dr. Brooks has written a recent article with several prominent co-authors entitled How the Science of Radiation Biology Can Help Reduce the Crippling Fear of Low-Level Radiation. It was in the May 2023 issue of Health Physics. In any case, I'm very pleased to welcome Dr. Tony Brooks to speak to us today, a fellow WSU Emeritus faculty member, and one who's helped, I think, elevate the reputation of our institution.
Thank you. I figured once I got so old that people might not want to hear me anymore, but I'd a lot rather be seen and viewed. And so I'm to that stage of my life where lots of my friends are being viewed rather than seen. I appreciate the chance to see all these young people in the audience. You're the future. We're the past. And it's really exciting for me to be here and talk. This is what I invested my life trying to do. So that bottom thing, just that I have to do that for some talks. Now, this is kind of an abstract of where I'm going to lead you through today rather quickly. What are the radiation effects from cancer and genetic effects? When I first started this field, genetic effects were thought to be just about the same impact as cancer. That's the second thing there. Does radiation increase genetic load? In other words, if we get radiated every generation, will it accumulate wipe out the species. And those are the kind of talks that I was hearing when I was your age. Don't want to wipe out the species with genetic load. Then the thing that I spent most of my life with is, what about material that's deposited inside of you? Inhaled, ingested, wounds. How hazardous is that? So we're going to talk a little bit about strontium-90 and yttrium-90. Strontium is a beta emitter that goes to the bones, and it stays there for a very long time. So once you get some of that, it's with you. Then thyroid cancer from iodine-131. Now, I know that I come from Kennewick, Washington, and we have a lot of money flowing into that community because of the waste. Now, they're not so much worried about iodine-131. It decays too fast and it's gone. But what about iodine-129? Very, very long half-life. It's going to be around for a long time. What about that? Thyroid cancer from iodine. When I was young, they used to say plutonium-239 is the most hazardous substance known to man. I thought, well, I'm going to look at that. I'm going to spend some time looking at the most hazardous substance known to man. Risk from liver, bone, lung cancer, all from plutonium. And the final thing, I think that there are a lot of costs that are being charged each of us because of the fear of radiation, low doses of radiation. So that's kind of where I'm going to go. It looks like I've got some pictures there on some other people. Can you guys hear me? They're on that? I guess the pictures don't move, but anyway, they can hear me. Okay, good, good. Okay. Now, nuclear weapons were really an interesting part of my early life. My hometown, St. George, Utah, was just over that hill right there. You could get up in the morning and go up on top of the hill, look out on Jackass Flats, and see a nuclear weapon go off. Boy, that was exciting. I thought that's really neat. And this is my hometown, St. George, Utah. A weapon would go off, the whole sky would light up above the black hill. And then we could count. And then we'd tell how far out it was that they set that thing off in Jackass Flats. That was really fun as a 10-year-old. We could do all of those kinds of things. You can see the cars there. I ran the service station there, so that was my job. I remember one time there was a fallout cloud. They stopped a bunch of cars and brought them into my station. We'd like you to wash these cars for us. Wash the cars for you? You mean you're going to take the radiation off all these cars and put it in my sump where I work every day? That didn't sound like a real bright idea to me. But I was a young kid. What would I know? So we did wash a lot of cars, concentrate the radiation right there where I was working. Now here, a lot of people don't realize how many atomic bombs we popped off in Nevada. Over 100 atomic weapons were shot off in Nevada above ground. A lot more than that shot off underground. But the ones above ground, the fallout clouds, didn't want them to go to Los Angeles. We'd wipe out a few of those Utah people. That doesn't hurt much. So they'd shoot them so that the fallout cloud would go north, and the national security site, we used to call it the Las Vegas testing ground. But there was one shot in particular that we called Dirty Harry. It took off, hit the Grand Canyon, ran up the Grand Canyon, and dumped in St. George very quickly. And so we had more dose than they had at the Fukushima nuclear accident the year so where it was. So we compared that one time. What was the regulatory action for those two things? St. George, they had a little truck with speakers on it. There's going to be a basketball game tomorrow night. Go in the house, there's fallout over the city. Crap, I'm playing ball. I'm not going in the house. If it's outside, it's going to be in the house. So that's kind of where I got introduced to fallout and radiation. So I did my master's degree chasing fallout through the ecosystem. It was in everything. It was on everything. Cows, the milk, the beet. It was everywhere. So when I went on to Cornell to go to school there, I thought, well, it's on everything. Is it doing anything? Huh? Are there any adverse effects? Am I going to have abnormal children? Genetic effect from me? Well, you know, they've got my genetic background anyway, so there's probably somewhat of a problem there. But I don't know how much the fallout would add to that particular problem that I had. One nice thing about it is that we marry women that are way better than we are. So our kids have a chance. Anyway, Dirty Harry hit St. George, and we wrote a little paper comparing Fukushima with St. George. We had way more dose than they did. What did they do? Told us to go with the house. What did they do at Fukushima? Practically wiped out the whole economy. So I'm the product of that, folks. And this is the Nevada. You can see one little hot spot right by where I lived. This is cesium deposition, Nevada fallout, worldwide fallout. And then this is the interesting thing. This is cancer frequency, okay? Now look at Utah there. Blue, blue, blue. Lowest in the nation. Huh, fallout must not have done a whole lot. What about the Mississippi River? What's going on there? Any ideas what's going on in the Mississippi River? Everybody in Chicago pees in the Mississippi. Okay, there's lots of stuff dumped in there all the way down. It actually has an impact, as you can see. And here's Utah's that bottom one. Okay, so the cancer rate in Utah is lowest in the nation. Oh, I wonder why that is. Lifestyle, folks. You take the cigarette and the alcohol out of a person's diet, you've really helped them out. A lot of people belong to Mormon church there. We don't use alcohol and cigarettes. Woo-hoo. So those are probably way, 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 way more hazardous than fallout. Okay, now we have a lot of data. Dropped a couple of bombs, didn't we? Got a lot of people exposed to some very, very high doses. In fact, they killed most of them, didn't it? The ones that survived, we've carefully followed each and every year for their whole life to record why they died. So we have a lot of information on radiation-induced cancer, between 1/2 and 3 sieverts. Now, do you guys deal with sieverts? Do you deal with rads or rems or sieverts or becquerels? I think what happened is people started to understand rads and rems. We thought we better do something else. We'll call them sieverts and becquerels. Then we'll be way smarter than they are. So anyway, they changed the whole system. So if you understood rads and rems, you might not have understood sieverts and grays. So environmental exposures are in the millisievert range. Background cancer frequency, about 40% of us develop cancer, 25% of us die of cancer. So what's the increased risk from a millisievert? Not much. I'd say damn near nothing. Twice damn near nothing is still damn near nothing. 10 times is still not much impact. Early studies with fruit flies suggested that the genetic risk might be almost as high as the cancer risk. But when we started doing mice, looking at the atomic bomb survivors, genetic risk is really almost insignificant. Fear of radiation-induced genetic damage should not exist. Does radiation increase the genetic load? In other words, if I got exposed a lot when I was living in St. George. Then I went to Cornell and studied radiation there. Then I went to Pacific Northwest Lab, Loveless Inhalation Lab. I got some radiation in all of those places. I'm still alive, dang. This is the Russell study of the megamouse study, as we called it. They would irradiate the males. They had to fractionate it, 3 plus 3, and they'd kill them. They gave them as much as they could. They'd still have some sperm left to cross with the females. 119,326 offsprings, 111 of them had mutations. So it became obvious mutations are not a big deal. Okay? And there's been lots and lots of studies since then that show these same kinds of numbers. Oops. Guy named Jake Spalding at Los Alamos National Lab decided, well, I wonder if the radiation increases if we keep getting it every generation. So he designed a study where he'd irradiate the male mice with 2 gray, which is about all he could do, and keep them all nice and fertile. Every generation, they'd cross them with females. The next males would be radiated and crossed with females. Did 20 generations, tried to look for anything that looked like a genetic effect. Looked at litter size, sex ratio, anything. Nothing. Just kept going. I asked him, Jake, how come you stopped at 20? You could have gone to 100. Well, the funding got cut off after a while. So that's why a lot of times we stop our research. But anyway, we were at a meeting in Philadelphia. Jake came in the next day, says, well, I went out and got stoned last night. I thought, Jake, why did you tell us your problems? Well, he went out and some people threw rocks at him. He kind of got out in kind of a rough area and people threw rocks at him. So that's how he was stoned. Now, internal emitters, this is what I was interested in because I knew I had a body burden. I could crawl in the Geiger counter that we had at the University of Utah. Oh, boy. I was one of the highest ones we had. I drank a lot of milk. I went on all the trips, gathering samples. Here, take some of this home to your sweetheart. So, I don't know, you graduate students now are probably a lot richer than what I was when I was a student, but a little meat and a little milk and some other things from the farm tasted pretty darn good. So anyway, I had one of the highest contents of both strontium and cesium because of my lifestyle. Eating off the land, you might say. So then I went to Loveless Inhalation Toxicology Research, and that's what we were studying there. We would have animals inhale about every known isotope. Strontium, cesium, plutonium, americium, just on and on. So what are the risks from inhaled strontium? Bone cancer. You do get bone cancer if you inhale too much strontium. There was a guy named Ernie Sternglass that was going around telling everybody that strontium was going to kill all the babies. I remember Busted was the director of Pacific Northwest Lab. After this guy gave his talk, he says, you know what we've just heard? When you cross an abalone with a crocodile, you get a crocobaloney. That's what we just heard from Sternglass. It's a crocobaloney. So those you may know, Leo Busted, he was really something. Radium dial painters. We know that there is a lot of cancer in those. Risk from iodine-131. Is plutonium the most hazardous substance? And risk from hot particles. So that's what I spent quite a lot of my time doing. This right here is bone cancer from beagles at University of California, Davis, that were fed strontium all the time. The pups were in utero after they were born. So there's the dose rates on this side, the time on that side. You can see up to about 1,000 days, nobody had any extra cancer. Very high dose rates, grays per day, that'd be 100 times that for rads, and time, 10,000. So you get way back there, at the end of their life, the dogs started developing bone cancer. And the radium dial painters, to apologize, this got shifted somehow. Those are supposed to be along the baseline instead of below it. Anyway, until they got up to 1,000 centigrade or rads, nothing. And those that were really sucking on the brushes, trying to make them just right to paint with, they started developing bone cancer late in their lives. Now, iodine-131. This was the main dose from fallout. Short half-life, very biologically available, goes right and sticks right in your thyroid. So now, out at the Hanford site, we're cleaning up iodine-129. Got to get all that iodine out of there. 8-day half-life, 10 to the 7th year half-life, okay, for 129. It's going to be there, folks. But this next thing, iodine-131 gives lots of zeros, times as much radiant per unit time as the same mass of 129. 1 microgram would give off the same ratios as a kilogram of 129. I don't know anybody that could put a kilogram of iodine in the thyroid. The thyroid's not that big. So to be cleaning up, spending lots and lots and lots of on cleaning it up, doesn't seem like a good use of my money and your money. So the mass is important. So there's been a lot of studies on thyroid cancer induced by a lot of different ways. We know that there was an increase in thyroid cancer after Chernobyl. I went to Chernobyl. A little town of Pripyat sits right next to Chernobyl. They did not tell the people in Pripyat that they're having a problem for about 2 weeks. So the children there got some horrendous iodine-131 doses to the thyroids. Oh, by the way, we're leaking an awful lot of radiation out of here. Maybe we ought to move all of you out. So that's what they eventually did. Too late, folks. There were 7,000 cases of thyroid cancer among the children at Chernobyl. Of those 7,000, there were less than 10 deaths in the 7,000. So if you're going to get cancer, thyroid cancer is a pretty good one to get. Just go in and lop it out and be on thyroid. You know, I had an interesting experience. I went in to have ear surgery one time, and the nurse came up and says, "Hey, it's time for your thyroid scan." I said, no, I don't need a thyroid scan. I'm here for ear surgery. Oh, no, I've got it right here on the chart. Get in the thing, take it down there. So they run me down to the place. Said, by the way, how much are you going to give me? Millicuries. I said, that ain't a scan. That's wiping out my thyroid, folks. I'm out of here. So I jumped up and run back upstairs with the nurse chasing me all the way, get me back in the wheelchair so I don't hurt myself. But they were ready to wipe out my thyroid. And I says, why? Well, as we palpated your thyroid, it was a little asymmetrical. Says, I want some more thyroid squeezers. Let's get some more doctors in here. So we got a bunch of them in there. They all, I think you're all right. It's only lasting now till I'm 86, so it's probably okay. Who knows? I might develop it late in life. So there's really no potential for increased risk from 129. Now, plutonium is the most hazardous substance known to man. A lot of times when you inhale plutonium, it's just small particles. These particles go and they deposit in your lung. So I decided, well, I'm going to measure chromosome aberrations in the liver. If you inject plutonium, it goes to the liver and the bone. So I had liver. We injected them with all these different kinds of radionuclides. As you know, the liver cells don't turn over, man. They just sit there. But if you go in and lop out half the liver, do a partial hepatectomy, the rest of it takes off. So this was ideal. These were Chinese hamsters, 22 chromosomes. My boss wanted me to work with dogs. 78 chromosomes? I'd a lot rather work with 22 chromosomes, 78 chromosomes. So we worked Chinese hamsters. But look, americium, plutonium, californium, about the same. Beta gamma's protracted, either with cerium or cobalt-60. Down here, about 20 times less than alphas than acute exposure. So this is one of the reasons that I got to go and, well, I'll go on this a little. Anyway, what if a single particle deposited? Cells sitting next to those particles are really getting the heck kicked out of them. It's a hot particle hypothesis. If the dose to those cells is high enough, it should produce cancer. So you can see here along these little stars that you can see, those are plutonium particles giving off alpha. And so we've got an autoradiograph. You can see that some of the cells that are sitting next to those particles look like they're really, really getting it, huh? Look right here. Those cells are really taking it in the air, aren't they? So if the hot particle hypothesis was true, these particles should be way, way more effective in producing cancer than the uniform distribution. So here's what I did. I made some autoradiographs. I went down to Otto Robbie, our aerosol scientist, and said, Otto, I want a bottle full of big particles, a bottle full of medium-sized particles, and a bottle full of little particles, and then I'll use plutonium citrate. I'll test the hot particle hypothesis with chromosomes and with cancer. He said, Brooks, you act like you're ordering a loaf of bread. That is a very difficult task that you've asked me to do. I says, I know, Otto, but you're really smart. You'll be able to do it. So they invented a centrifuge where they put the particles into the centrifuge, spin it, the big ones would hit the foil early, the little ones would go down. So we just cut the foil off, resuspend them, monodispersed particles, all big, medium, and little. So here's the citrate. You can see the little tracks, 0.3 microns, 0.4. You can see that's liver cells. So the guy sitting next to that, they're just getting a lot. Now, when I did the chromosomes on this, I expected to see a very unusual distribution of damage. I expected the big particles would produce a lot of damage in a few cells. The others would be rather uniformly distributed. I don't know if any of you have done an experiment where the Chinese hamsters didn't read the protocol, but they obviously didn't read this one, because that's not what happened. Chromosome aberrations per cell, dose, different particles. You can see the 0.8 micron one way up there at the top. It's right next to the littlest ones. The citrate is right there. Okay? All on the same line. It depended on the total dose to the organ rather than the localized dose, which blew me completely away. It's chromosome aberrations were supposed to be caused by a particle hitting the chromosome and breaking it. Almost nobody hit the chromosome with those 0.84. The ones that got hit, they really got hit. And so the whole organ was responding as opposed to the individual cells, which to me, I still haven't quite figured out why the devil that happened. But one bad thing about it, you do science, you have to believe the data. We have preconceived notions a lot of times when we start a study. Data says, no, your preconceived notion was wrong. The hamsters did not read the protocol. And this is the cancer data. The citrate was the most effective, all the particles there, and the controls. So the citrate had the same total activity, but way more cells exposed. The particles had very different numbers of cells exposed at different levels, but the liver responded the same. Again, I almost worried about publishing it. How would I explain that to the editor of the magazine, the journal? But it did get published. So, hot particle hypothesis not supported. To get cancerousness, to expose as many cells as you can to the radiation. If you only expose a few cells, like the particles did, it's not as effective as the citrate. Uniform distribution is more effective than any other alpha emitters. They actually published it. I don't know why it's more effective. I don't understand how the devil that happened, but them's the data, folks. Now, what about radon? Have you got radon in your home? Is it killing you? Radon produces the biggest dose from the environment than anything else, way bigger. What if radon is killing us in our homes? Now, this is the one time I got to go and talk to Congress. They were passing laws on how to mitigate radon. And we had the senator from California there running it. He said, Dr. Brooks, I'm not interested in your science. I have a wife and 4 children that I dearly love. If I had the radon level recommended by the EPA in my home, what would you do? I looked at him and says, well, you know, I have a wife and 6 kids. He says, well, do you dearly love them? Well, 3 of them are teenagers. That brought the house down anyway. But then I went on to explain that the radon levels that they've got set are so low that if I had that, I would do nothing. If I had 10 times that, I'd open my windows. I'd put a fan in the basement. It's easy to solve the problem. Evidently, that wasn't what they wanted to hear, because they went ahead and passed lots and lots of registration, set up a great big department in EPA to study radon. Anyway, it shows how impactful science is sometimes. Let me go back and just look at that just a second here. If you're a never smoker, here's your lung cancer rate, deaths, 1950, 95. If you have radon and you mitigate, you change it that much. Now, if you're a smoker, your risk goes from here up to there. And if you have radon, woohoo, another 60,000 lung cancer deaths. So what's the bottom line? If you have radon, for heaven's sakes, don't smoke. Because you've already raised your risk way up to here. You add radon to it, wow. But if you're a never smoker, so if you've got radon in your house, please stop smoking. And if you've got a smoker in your house, you might encourage them to go outside to do their smoking. That's as simple as that, see? So it's not the second or third cause of cancer. Interacting with cigarette smoke, that's a big one, okay? If you have radiation in your home, don't smoke. Now, the last quite a few years of my career were funded by Pete Domenici. He was a senator from New Mexico. Senator Domenici said, the Energy and Water Appropriation, we initiated a 10-year program, $13 million a year. And we will be able to understand how radiation really affects systems, really what is going on. For the first time, we'll develop protection centers that are based on actual risk. The program lasted just over 12 years. I ended up working out of Washington State, but I was the chief scientist for this program. So we set up all the reviews and did all of those kind of things with it. Wonderful program, lots and lots of good data on low doses of radiation. See, always in the past, you want to have something that you can see a big change. That way you can send your paper out to Science or Nature or some of those places, huh? God, if you can't see anything, who's going to accept that paper? Anyway, that was my job then for the last 10, 12 years of my scientific career, was to be the chief scientist for this. I got acquainted with lots and lots of good scientists through this. These are some of the biological responses that we didn't know about before we started the program. Genomic instability. We used to think it was a single mutation. Now you'd radiate them. They'd go through quite a few cell generations, and then up would come the mutations, the aberrations, the genetic damage. Bystander effects. That's what my plutonium particles was telling me. The whole organ responds as opposed to exposure to individual cells. The organs are responding, not the individual cells. And you go down to low doses where almost nobody is hit, you still see these responses. Now, what about those responses? High doses, they're almost all damaging. Low doses, a whole new set of genes gets turned on. A whole new set of proteins gets activated. Most of these are protective. Oh my goodness, you mean a little bit of radiation is actually good for you? That was really hard EPA to accept. Anyone who set standards, how could you say that, Brooks? You must be an idiot. And you might have been right. Adaptive responses. new little agouti beasts turned out to big, fat yellow beasts. And these yellow beasts were very cancer sensitive. He said, oh, man, what we do if we radiated them at those same times that we're treating them with this chemical to get this effect? Oh, I wonder what would happen. So we funded him. He ran the experiment. He and I were driving home from Washington, D.C., and in a cab, his phone rings, talking to his tech. Damn, brown, brown, brown. They're all brown. What do you mean, Randy? Well, the radiation, instead of making them yellow, made them all brown. I said, geez, that's wonderful. Why do you think he was irritated? Huh? Come on, you guys fight for money. If he would have found a new effect of radiation that affected offspring when the mother was radiated, he'd had money poured on his head like he wouldn't know what to do with. Brown, brown, brown. Dang, they're all brown. I took him to a National Academy of Science meeting, and he presented his data. God, they just jumped all over him. I said, wait a minute here. Wait a minute. It's all right to jump on somebody's data, but you don't need to get personal about it. This is what Randy did. These are his data. If you don't like them, I'm sorry. But the mice didn't read the protocol the way you did. Dang, they're all brown. So you see the dose here? Those are rads. They start off about equal number of each. About 2.5 rads. God, they're all brown. Anyway, Randy was not pleased with his results. And he took a lot of crap from a lot of radiation protection people over his data. So don't get angry with Randy, get angry with the mice. They didn't read your protocol. This is what they do. So this is kind of a summary. Made us make many paradigm shifts in radiation biology. Hit theory, it used to be bang, you hit them, bang, bang. No, bystander and tissue effect. The whole tissue is responding, not single cells. Linear dose response versus protective adaptation. We set all of our standards on a linear dose response, right? All the way to 0. No, you get down here, you're supposed to drop it off. You mean I'm not important? I work for the EPA. We know that any exposure to radiation is going to cause a detrimental effect. Boy, they jumped all over him when he presented his data. Mutation theory versus genomic instability. A lot of times you radiate them, then quite a few cell generations later, you get this. Now, this is what bothered me a lot. This is right here where they quit funding us. The funding agency said, no, we don't want to fund you anymore. I thought, how come you don't want to fund us anymore? You're finding out things that we don't like. Maybe not, maybe. But anyway, my job was terminated. And so here I am, almost an old man, working at Pacific Northwest National Lab. I lose all my funding. I'm sorry, you're going to have to be out of here in 2 months. Oh, geez, come on, I've been here forever. 2 months. So what I did is I sat down and wrote grant proposals. I wrote them to DOE, I wrote them to NASA, I wrote them to EPA. Never in my life had it happened. 3 of them got funded. Woo-hoo, I had some money. So I just walk across the street to Washington State University. Hey, we love you. You got money. We will give you an office. You can even have a secretary. Woo, woo, woo. Anyway, that's what happened to me. Low doses are nonlinear at all levels that we studied. Molecular, cellular, tissue organization, people still aren't willing to say it works in humans. Those are just animals. Those are not humans. The science suggested that linear no threshold hypothesis overestimates risk. Well, that's all right. I guess that's what they say. Well, that's all right. We'd rather overestimate it than underestimate it. Well, yeah, but you're spending millions on it. Go over and watch them clean up the Hanford site. Whoo-hoo. Serious money. We love it there in the Tri-Cities. That really runs our city. So the dinosaur of the linear no threshold hypothesis is scientifically dead for low dose risk assessment and overestimates risk. However, it remains used. Is it useful? So I've got a bunch of guys throwing at the linear no selective apoptosis. Damaged cells just kill themselves. Bystander effects, ROS modification, adaptive protection. Those are going to knock that. And we were just starting to launch these at them. Epigenomics, proteomics, metabolomics. Then we could start maybe getting down to where we really understood what was going on. You're done. We've cut you off. Radiation's a poor mutagen and carcinogen. In the low level may even be protective. Low dose and dose rate risk is very small and difficult to detect. Fear of radiation kills people. Now, how does it kill people? Lots of times people go to the doctor and they say, we need to take an X-ray, a CT scan. You're not going to radiate me. So they refuse the very thing that they need to diagnose their disease. And that's, well, and that's very expensive, like I say. Cleaning up the Hanford site is quite an expensive job. So here's some of the costs. Nuclear waste, 2 to 4 billion a year. Try to clean up down to levels about background. Way lower than Salt Lake City, way lower than Albuquerque, New Mexico, way lower than Denver, Colorado. Come on. We can't see any increase in cancer in those cities. They're not really cleaning anything up, they're just moving it away. They just move it from one site to another and try to get it further away from the river. You know, it's really interesting because I was on the Hanford Advisory Board. We were talking about this. I raised my hand and said, you know, you guys are monitoring the wrong side of the river. The radiation coming in from the Hanford site is way less than the radiation coming in from the other side where all these big farms are. They're using 1616, potassium 40, coming in by the becquerels and gigarels. I mean, it's just flowing in there. You got your monitors on the wrong side of the river. Of course, the people at Hanford Environmental Science, they didn't like that very much. I only lasted another couple of years. You're out of here, Brooks. Anyway, nuclear power. It looks like to me that the world is starting to build nuclear power plants. Everywhere in the world, China's going crazy with them, okay? You give China a cheap source of energy, all of their other resources, they're going to kick us around the countryside economically. I don't have to tell you that it's clean and no greenhouses and reliable. But in medicine, a lot of people aren't getting the proper diagnosis. Cancer, radiation is almost essential in all kinds of cancer cleanup. So that's what I'll end with. Thank you very much for your attention. If you have any questions, you people on the Zoom evidently can ask them also. Go ahead.
So I'm interested in that radon smoking would really have a
Right.
significant effect. So if you regress cancer, yes or no, on a host of independent variables in their interactions, one of them could be smoking, whatever. What kind of interactive effects do you see? Is it just smoking and radon? I mean, I'm sure there are other interactive effects. And then, you know, low dose radiation, how much does it add to the cancer risk? I mean, you know,
Yeah.
other than radioactive effects. I mean, I understand you presented the data from Utah, you know, all blue, the Mississippi Valley all red, and there may be some larger effects that mask the small effects that low dose radiation can.
Right. Now, we know that there's a lot of chemicals in the Mississippi River. Whenever they have a storm or something down at the bottom of the Mississippi, what do they stir up? A ton of junk, right? A lot of which is carcinogenic, okay? So as far as interactions, they know that radiation and smoking interact, okay? Radiation and alcohol interact with liver cancer. One of the hard things about taking the Hiroshima and Nagasaki data and applying it here is the diet. Japanese have a very different diet than we have here. In fact, they eat some things there, bracken ferns, that are just loaded with carcinogens. And so if you try to take their data and move it here, God, our diet's very different than theirs. And so all I can say is that I thought we were just right on the cusp of making some real serious advances. Got cut off. Now, it has been reinstated. DOE has taken the money that's been granted by Congress. I have not followed up where the grants are. But if I was a student and was interested in radiation, I'd probably talk to DOE. Say, hey, I understand you're initiating a new low dose program, and then you have lots of money to do this. Could I have some? This is my idea, you know. I think that I know enough about this particular area that I could be very helpful. And hopefully you can get a grant that way right now. So, yeah, but the interaction is really a tough problem. You get so many different things. I tried doing that at P&L, combining this and that. God, there's so many combinations, it blows up on you. Blows up on you really quick. So I took some of the organics that are in the tanks, combined them with radiation to see what I could see. Not much. Okay, I tried this, tried that, not much. So it's, I guess cigarette smoking causes enough inflammatory response that I think, anything that causes inflammation increases your risk of cancer, okay? And the smoking, you see, smoker's lung. I worked at Loveless Inhalation Toxicology Research Institute where that's what we did, is look at lungs. Smokers, non-smokers, radiation, we radiate them with everything, so on. So yeah, it's a real problem.
But it does exist.
Oh yes, obviously, obviously. And I think radiation gets credit when credit should be someplace else. How come you always blame me? What about this? What about that? What about this? All very hot known carcinogens. Okay, but easy to blame radiation.
Tony, there are some new treatments that have been developed in medicine that are making use of
Just like you to comment about
variety of isotopes.
regulations. I noticed in the book that you mentioned that the French Academy of Sciences has now accepted the idea that things are different in the low dose region,
Right.
but that hasn't happened yet in the United States with the National Academy.
Right.
Could you make some comments on that?
You know, I took Randy Girdle to the United States Academy of Sciences, and he gave the paper on the yellow mice. God, you wouldn't believe the anger that stirred up. I says, God, don't blame Randy, blame the mice. They did the experiment. He just showing you the data. But there are so many people in this world that make their living making sure that we don't get any radiation. It's a tough sale. We took a bunch of our scientists to the head newspapers in the Washington, D.C. area. They brought all of their editors in. We showed them some of this data. I can't get that in my newspaper. If you would have shown that radiation was more hazardous than we thought, front page, less hazardous, oh, gee, my editor might stick it at the end of the newspaper somewhere. But that's a fact of life. And so they're trying to sell newspapers, right? Anyway, so that'd be my only comment on that, is that we tried very hard. We got all the regulators and the scientists and the bureaucrats together. Regulators give their talk, everybody else sleeps. Bureaucrats give their talk, everybody else sleeps. Scientists give their talk, everybody else sleeps. My boss said, that was a complete failure. I says, no, that's a start. We've got to start helping these people talk with each other so that the regulators know the recent science. Anyway, we were only able to do that one time at one of our meetings because that was a waste of time. Go ahead.
So you talk about how there's a real societal cost to fear of radiation. As the younger generation looking for guidance from the older generation, what can we do to mitigate that?
You know, I think every generation, a lot of people that are sort of your age don't even know about Hiroshima and Nagasaki and the atomic weapons, you know, and the doses from radiation there. They don't know about the Hanford site and all the radioactive material that's there. So every generation has these same questions. What about diet? What about this? What about that? All very important, and I think worthy of some research. I don't know if that's what you're asking me or not. Right. I think that you've got to have a database to start with if you're going to steer any. And you have a set of regulations that are knocked in stone, basically, almost. Trying to get those out and new regulations in for the low dose region where almost all of our exposures occur. Very, very difficult problem. I can say we tried mix and match, and we tried editors, we tried a lot of different things, but communication is really hard. When you try to talk science to the general public, they might keep their eyes open for 20 minutes if you're good, but usually you'll have most of them asleep by the time you're done. The same way, if the regulators try to talk to the scientists, they speak different languages. I worked at DOE headquarters for a while. I had to learn a whole new language. Okay.
Tony?
Yeah.
Okay, I really enjoyed your talk, by the way. One thing that struck me is that in medicine now, they are moving to a lot of radiation treatments for an awful lot of products. I've had PSMA procedures done to identify where prostate cancer is in my own body. And I know that they're marketing other products to try to destroy those cells once they've been identified, but they're not using low doses. So there's kind of a disconnect. The low dose issue that you're talking about, I think, is very real and overblown in many ways. But yet people are looking for high dose solutions. I guess I would call it that. I don't, probably wrongly, but it would seem that that's where they're moving. I mean, what are your thoughts on?
Well, you know, like you, I had prostate cancer, okay? 2 gray a day, 5 days a week, 9 weeks.
Yeah, that's what I had.
They killed everybody in sight. I shouldn't even be able to urinate by now. But by focusing it carefully, they were able to hit the prostate gland and the other functions of the kidneys and still work. Okay? And so, yeah, high doses are really, really essential in a lot of cancer therapy. And this low dose thing, I think, is more a regulatory problem than a medical problem. You know, why do we spend so much money trying to clean up to a level that's below Denver and Albuquerque and Salt Lake, you know? That's my big problem with it. I really agree with that high doses are really essential. Made a little mark or two there and focus in on that sucker. 5 days a week, 9 weeks, 2 gray a day. That's a lot of dose.
You
Well, I
mentioned,
don't know whether you had the procedure I did, but I had the procedure where they did a full body scan and knew exactly
about
where the
other
cells were.
effects, lifestyle effects, all the other things that contribute to cancer. And yes, that's certainly true. Uh, in all these studies, there's an underlying assumption that the lifestyles are the same between those with and without the study groups. Now, any randomized controlled trial has the trouble that people who tend to participate in these studies could be in some way fundamentally different from those who don't.
Right.
And so transferring from the study to the general population is a very big different than comparing between the study populations where we do control for the lifestyle effects. And so to a little bit, I think your statement about how we need to look at all these other lifestyle behaviors is a little disingenuous in deciding whether the studies on low dose radiation are valid or not, or whether it's a problem. We are always looking for the marginal effect,
Right.
not the overall effect.
Amen.
Other than that, I very much enjoyed your talk.
Well, you know, I guess each one of us kind of focus on what we did. You know, I'm sure that there are people who do farming here that could give a talk that would really educate me very quickly. But this is what I did. And so that's kind of why I focus on that. But when you try to move the Japanese data to the United States, it's very, very difficult. Their diet, their lifestyle, everything is way different than ours. And so you take this data, the A-bombs, and try to move it here. There's really a lot of other things that you have to put in that can't be put in. People are a terrible experimental animal, okay?
We'll
Every
take,
one of us.
take two more quick questions.
Okay.
I was just wondering if you could comment on magnitudes of effects. You're talking about low dose radiation exposure. How does that magnitude compare with the variability in natural radiation exposures that you get to in various parts of the US or in the world?
Yeah, that's a good question because I lived in Albuquerque, New Mexico, okay, for 20 years, about 4 times the background rate that the people on the coast get. Okay, and so this low dose is down in that range, okay, about 3 or 4 times the low dose would be Albuquerque. Okay, the Sandia Mountains are 10,000 feet. The city is about 6. And so they got a pretty good dose.
Thank you for your talk. It was very enlightening for somebody who I don't know anything about biology. So you talked about this experiment with irradiating mice, and that their offspring didn't have any mutations or cancer. Do you know if this study has been done on mice, on both female and male mouse that were irradiated, and what's the difference between the female being irradiated rather than just the male, or both parents being irradiated?
Yeah. Well, the female, the oocytes are really quite sensitive to radiation killing. And so if you radiate the female, you make her sterile. Okay? Those doses that they were giving for the males would sterilize the females. So if you try to do that study, you're doing a much lower dose. And so if I'm a funding agency and you tell me I want to radiate a bunch of female mice at almost nothing, I might tell you, I don't think I'm going to give you any money. But it's a good idea, see, because there's such a difference in sensitivity between males and females to reproduction. You can sterilize women really easily with low doses of radiation.
So that means that women that have been irradiated in nuclear sites or like Chernobyl sites will rather be sterilized than have mutant kids, right? So that's a good outcome, I guess.
I don't know that, how would you do such a study?
I wouldn't say that I would do it, but I'm just saying that in the cases, in like the historic cases that we had.
Yeah, right. I
women-
appreciate that comment because it's a really valid one. I need to sit down.
You earned it.
My name is Nick Loveridge. And my friend Bill Budd was on TV and he was down at, he's an environmental scientist. I was watching you today and he asked that